I think we have two more talks this morning. The first one is structural neurobiology, the dense reconstruction of neuronal circuits.
Thank you very much for the invitation. It's a great pleasure to be here. I want to pick up where Jeff Lichtman left us yesterday, which is the topic of connectomics and what we and others are trying to do to densely map neuronal circuits. The work that I'm going to show is mostly back from my time as a postdoc with Winfried Denk at the Max Planck Institute in Heidelberg. I have now my own group in Munich at the Max Planck Institute, where we are continuing these efforts, as I will show at the end. Now, starting again with the brain as the goal of, or as the subject of imaging, Jeff provided nice arguments why it's interesting to look at brain structure. I want to add another one that goes like follows. All of us have 85 billion neurons in our brains. That's the latest number. It has a standard deviation of something like 5 billion. We have no idea what that means. But let's say 85 billion each of us. That is very impressive. And you could say that is already enough to justify why the brain can do things that other organs cannot do. But then somebody comes and tells you that the liver, or as we know, the liver has also many cells, right? So also several billion hepatocytes. So in that sense, it's not so amazing that the brain has so many cells if an organ as stupid as the liver can also have so many cells. What is really the difference is the nearest neighbor relationship of the cells in these two organs. So if you think about a hepatocyte, then it directly interacts with other hepatocytes that are located directly next to it. So the nearest neighbor interaction is with a few neighbors. In the brain, however, as we know today, a single neuron interacts with, on average, 1,000 other neurons on that scale. These are numbers from mouse, where it's probably true. Man probably also. It may be more like 5,000, but the scale is a few thousand other neurons that particular neuron directly synapses with. And that direct interaction in a nearest neighbor sense is not local necessarily, because, as you know, neurons can span the entire brain to make their connections, but it is direct in the sense that it's a direct synaptic connection. And in that sense, you can describe the brain as a network with 85 billion nodes and an order of 1,000 or more. And that is really a scale difference to any other organ that we have, maybe excluding the immune system, but really all the other organs have a much lower order of graph connectivity, if you want. So, and this simple argument alone, I think, is enough to justify to really care about the brain's connectivity and to try to get there at mapping out the networks in our brains or in model brains. Okay, so how can that be approached? And here, just as an example, a circuit in the cortex of a mouse, where in a very prominent location that is layer 4 for those who care, there are a few thousand neurons, and all we know today about their connectivity, just as an example, comes from measurements where you stick in 2 electrodes at a time and measure the pairwise connectivity between nerve cells. That is kind of the standard method still to unravel connectivity on the electrophysiological level. And what comes out of such measurements are very important numbers, like the average pairwise local connectivity, which turns out in that circuit to be 15%, which means that a given neuron connects to on average 15% of the surrounding neurons. And that is actually a pretty typical number that you find in most local neuronal circuits, for instance also the olfactory bulb or other cases, where on average it's roughly 15 to 20% local connectivity. That is the local connectivity. The global connectivity, of course, is much lower because, as I said, a given neuron only connects to, say, 1,000 other neurons out of millions with animals or billions with human brains. Okay, so this is an important number, but it's not enough if we really want to understand something about the network structure of neuronal circuits. And this is illustrated in this slide where, just as a sketch, I've plotted 100 neurons and what you can call a connectivity matrix or an adjacency matrix in more general terms of the graph of the neurons that we're looking at here. And that is, works in the following way, that you basically have a presynaptic and postsynaptic axis, and all the blue dots here are supposed to be existing synaptic connections, while white is supposed to be no synaptic connection. And the point here is now that the matrix up here has a filling fraction of 15%. So this is, in a way, one incarnation of a circuit with average pairwise connectivity of 15%. And the top matrix is really just random in the sense that there's not anything more, or there's no more structure than that random pairwise connectivity. However, you can picture another case where you have two cell assemblies that highly connect within each other, but very sparsely so only between each other. And this would be the connectivity matrix. This would be one of the possible graphs. Now, the point is that the filling fraction of that matrix is also 15%. So if all you can do is pairwise mapping of connectivity, then you have no chance to distinguish a circuit as random as the one up here from one as structured as the one shown in the middle. And the same would be true for another model that has been suggested, which is Synfire chains. Again, you can construct a matrix with an average filling fraction of 15% that is highly structured, but if you just compare these average numbers, you have no chance to distinguish them. So this is really the problem, and pairwise mapping doesn't get you there, as I just illustrated. And in order to understand whether any of these circuit models or completely different ones are implemented in our and other mammalian brains, we have to measure the connectivity matrix. And that is the goal of the efforts we are following, and I want to show you how we do it. Now, why is this so difficult, and why is imaging the brain connectivity such a huge problem? Jeff has introduced parts of the or aspects of the problem. The way I would phrase it is the really difficult part is that some pieces of neurons get very, very thin. These are typically spine necks, for instance, or thin parts of axons. And thin means less than 50 nanometers in diameter. We know now that in certain brains, for instance fly brains, it's even thinner. It can be like 30 nanometers or 20 nanometers in diameter. So this is really, really small. At the same time, however, neurons span scales of at least hundreds of microns, if not millimeters, or even centimeters in the human case. So this scale difference, or this scale span between a few tens of nanometers to hundreds of thousands, if not millions of nanometers, is what has made it so terribly difficult to map neuronal networks, because you have to span these scales. You have to be high resolution, large scope. Therefore, electron microscopy is a technique of choice if you think about the resolution requirements. I just want to briefly comment on the light microscopic hopes that have been out there. The problem is that with conventional light microscopy, you have a resolution element on the scale of 200 nanometers, maybe 500 nanometers on a side. Now, in the most difficult parts of the brain, say the cerebral cortex, neurons are packed so densely that the neurites in such a resolution element are easily a dozen or more. And that now means that, at least with conventional light microscopy, even if you have several colors, you would need 12 or 15 distinguishable colors to actually image this kind of data just with light. So if you have mixtures of colors, it doesn't work. You would have to have 15 or something like that unique colors to resolve densely neuropil with light. And as long as you're not using a super resolution microscopy, this is basically not possible for the densest parts of our brains. It's nicely possible for sparser labeling, but for dense labeling, light microscopy cannot do the job today. So that is really an important point, I think. Okay, that is why we and others have focused on electron microscopy. How has that been conventionally done? Ever since the 50s and 60s of the last century, when electron microscopy was introduced into biology by Gray and Colonier and others, people have done serial section transmission electron microscopy, which means that you take a block of tissue, then you slice it up into very thin sections, meaning typically 50 nanometers or more. And then you have to take these sections and put them into the electron microscope, and then you shoot an electron beam at the sample and measure basically the contrast by the deflection of the electron beam, and then you have a picture here, and then you can align the sections. Now, that works very well. However, it has a very big problem, which is this step here, where you have to carry the sections physically into the electron microscope. So you're really cutting sections. They are very, very thin. And then you have to transfer them into the EM to get the picture. And that step is really difficult to do for a large series. And as you can see, if you have a 50 nanometer slice, you want thousands of these slices to get over sizable distances, which you need, again, because neurons are large. And this is such a tedious problem, and such a big problem, that this method of serial section TEM has basically only once been applied to an entire circuit, which is the famous example of the C. elegans nervous system reconstruction by Brenner and colleagues. They took 9,000 roughly slices. It took them 15 years, major effort, done once. What we're doing is a method that changes the process of cutting and imaging using just a basically very simple trick. So instead of taking the block of tissue, slicing it up and imaging each slice, what we do is what we call serial block phase scanning electron microscopy, which means we take the block as it is into the electron microscope chamber, and then instead of first cutting and imaging, we first image and then cut. So what we do is we take an image from the top of the block. This now, of course, has to be scanning EM because we can't shoot the electron beam through the block anymore. So we scan the surface of the block, and then we have a diamond knife installed into the electron microscope, and that diamond knife then shaves off, if you want, the top of the block. Then we take another image, scrape off, another image, scrape off. And this simple inversion of cutting and imaging into first image and then cut means that we take the image while it's still on the block. And that means that the images are very nicely aligned, and the distortion problem that used to be the biggest challenge when transferring sections into the EM is gone, because we take an image when the block is still intact. And the second big advantage is that this whole process is completely automated. So we can run these sequences of thousands and thousands of sections over long times, weeks and so, but it is not manual labor, but that of a machine. And that helps a lot. To illustrate that, we have a movie here. That's just a mock-up, so maybe you can see the yellow, in this case, electron beam scanning the surface of the sample, and then we have the knife coming from the left, shaving off the top, and then another image is taken, and the knife comes in again. And I should say that this is highly sketchy. The imaging itself takes much longer per slice because we take large areas, and maybe I can focus your attention very briefly on this cutting process. We are very happy about this animation because it curls, actually. Maybe you can see how, so we're very happy. It's difficult to get. Okay, so this is the technique. Now, how does data look? This is an example from rabbit retina, where you can see many neurites running around, and then out of this comes one of the reconstructions of just one of the more than 100 neurons in this particular tissue block. This is still a reasonably small tissue block, but already you can see how you can use this kind of very consistent data now to reconstruct neurons from it, and this is just one example of an amacrine cell from that particular block of tissue. Okay, so imaging is one major problem, but in a way, for at least these kinds of volumes, as I will show later, we think this is reasonably solved. What, however, and this is a focus of my talk, what, however, is the really biggest problem is reconstruction. And I think we've heard examples of that yesterday, and in my view, this is probably for large throughput imaging efforts, in most cases, the main challenge is not actually anymore the imaging, because we've become very good at that, but is reconstruction at the same scale. And I want to illustrate that. So in order to reconstruct neurons, like the one we've just seen, usually what you do is you contour the neuron in each slice and then create a volume representation out of that. Now, that works very nicely and looks very nicely, but it takes forever. More quantitatively, 200 to 400 hours per millimeter of path length. What does that number mean? Well, circuits that we're interested in typically have an integrated path length of at least meters, if not tens of meters. And that just means adding up all the path lengths of all the neurons involved in that particular circuit. So for instance, in the cortex, even just rat or mouse, in layer 4, the circuit I briefly introduced before, we're talking about 40 meters path length. Even the retina locally, it's half a meter, or 2 meters of path length. That is a lot if you think about the number of a few hundred hours per millimeter reconstruction time, which means that manual reconstruction adds up to these absolutely terrible numbers, like several million work hours. And this is really prohibitive, even if you have a dedicated lab and everything. So this is really prohibitive and means that reconstruction is limiting what we can do in terms of dense circuit reconstruction. What have we done to overcome this? The first thing we had to do was to develop software tools to actually be able to browse our data, because our data is easily terabytes, and at the time, and even today, it's not the usual kind of data size that you can load into RAM. So we built a browser called Knossos, which in a Google Maps style allows us to browse around in that data on any laptop. It loads data when it needs it. And importantly, we have 3 planes of viewing because our data is reasonably isotropic, and we can really navigate in all dimensions, and that's critical to follow neurites, as you can see in a second. And the second aspect of that tool is that we can reconstruct neurons not by contouring each slice, but by more like clicking along the center line, or if you want, it has more the aspect of flying, or at least freely navigating through the volume, and whenever you want, you drop another point, and by that reconstruct what that neurite is like. And how does that look in a more kind of rendered way? So this is now a flight along one of the axons of a bipolar cell, in this case in the retina, along such a skeleton that has been reconstructed. And this is just a movie to illustrate 2 things. One is, if you focus on the center of the movie, that actually this kind of data seems reconstructible if you get used to the weird colors or lack thereof. But also, what you can see is maybe in the next phase of the flight that if you look more at the surrounding of that very process, how complex actually our brains are, right? And this is just rabbit retina, so it's really a very, very dense mesh of neurons or neurites, and all of these different processes belong to probably other neurons. So this is really the problem we have. It's extremely dense. And by the way, the distance that we've just traveled until this point now is just 30 micrometers. Right? So it was a lot of stuff to look at to just travel 30 microns. Okay, this enhanced annotation helped us a lot. It sped up the reconstruction by a factor of 50, which really made a difference, as I will try to illustrate later, because with that kind of speed, we could at least start getting data from that kind of images. Okay, now speed, of course, is always only one side of the problem. The other aspect is accuracy. Right? If you have any method that analyzes data. And one test that we did was we asked 2 self-proclaimed experts to reconstruct the same neuron, which is shown here. They started at the same point in the soma here, and in green you can see the agreement between those 2 so-called experts. And in red, however, you can see the disagreement, which was also there. So there were several locations where those 2 annotators disagreed. And that was particularly puzzling because it was my close collaborator, Kevin Brigman, and myself who had annotated this, and we weren't happy about that kind of disagreement. And then we went back to the locations at which it happened and found that, this is just one example, that actually those locations were not really difficult to analyze. It's difficult to show in 2D here, but most of these locations were missed branch points. And by most, it was really like 12 out of 13 were missed branch points. And that was puzzling because it's not difficult to see these branch points. But if you think back to the movie that I showed, the problem we think is actually attention. Because in order to find all the branch points, you have to be constantly attentive and look at all this crazy data that's coming in and make sure that you pick the one branch that's going off from that particular neuron and not happen to jump into one of the other neurons. And that is now something that humans are notoriously bad at, continuous attention when doing a task like that, or basically in general. So, and that is really a problem because it means we can't even trust the experts. How do you solve something like this? Well, we've heard about that before, that one way of getting rid of such problems is just crowdsourcing. So instead of asking one person, you do a multi-annotator approach and ask not 1 but 2 people, or even 3, or even much more. And that allows us to now really sample decisions and then use simple decision theoretic approaches to calculate what is most probably the correct answer. So again, this is a setting comparable to what we've seen yesterday in a pathological setting, and actually that's one of the fields where this has been pursued already pretty extensively. How can you consolidate the judgment of multiple experts into something that is most likely true? And this is one other example here. So in reality, this looks like this. This is a reconstruction of 50 people So 50 students were asked to reconstruct the same neuron, and this is what they gave us. In the end, this came out. It's a mess. And then the algorithms we developed to consolidate these decisions allow us to then calculate a consensus, and this is a much more realistic reconstruction of an amacrine cell. So just as an example of how we're using, again, decision theoretic approaches to use the judgment of many people to get at a more correct answer. Okay, you may have been asking yourself, why aren't we just asking computers to do the job? Why this mess with, or this work with humans? And the answer is, as again, we've heard before, it's just one of these problems where computers are not yet good enough at. And more specifically, the task is to convert a grayscale data into a segmentation. You cannot do that globally because a decision at one point in the data set affects a decision at the other end because neurons are non-local. So usually what you do is you do a local classification first and then use some growing or segmentation algorithm to actually get a full segmentation of the volume. And that is something that nicely translates into a general machine learning setting, because basically you can say that you have a surround of voxels which makes up some reasonably high dimensional gray value space, and then the decision you want to make is usually pretty simple, something like I'm in a neuron, I'm not in a neuron, or it's a wall, it's not a wall. So it's a typical classification problem for which you can use all the machine learning methods that are out there. For instance, nearest neighbor template matching, or more complicated things like neuronal networks or support vector machines. We've tried all of this and worked with groups in the field and tried it, and it's actually pretty nice. So you can convert gray value data into more binary looking representations, and then you can use segmentation methods to get at pictures like these, which kind of look nice. It looks neuronally kind of, because these things seem to be traveling here through space. However, if you do more quantitative metrics on it, you find that there are many small objects that are not correctly assigned to any neuron, and quantitatively we found that all of these methods basically run against a wall of roughly 40 micron correct neuron reconstruction. And if we think about the length scales of a neuron, that's just not even close to enough. So if you break a neurite after 40 microns on average, that is actually millimeters long, then that's just no option. So the initial hopes of using some automated method, well-trained method to do the reconstruction broke down in the field. And therefore, instead of despairing, we decided to combine the manual approaches we have with kind of the automated approaches to actually do the volume filling. And I will show you the results of this next. Okay, so this was the methodological part. I tried to introduce the imaging we're using and the challenges of the reconstruction, the solutions to those challenges that we at least have been using. And now I would like to show you two examples of what we can do with this kind of approach. The first example is, both examples are from the retina. The first one is an example where we have performed a sparse reconstruction of a few neurons in a large data set. And by large, we mean something like 300 micron on the side times 60 in the third dimension. So this is a block of retina that we've imaged in the EM, and importantly though, we only reconstructed a sparse subset of the neurons that are functionally identified. I'll show you that story in a second, and then the second part will be about dense reconstruction. So for the sparse reconstruction, we were interested in a particular type of ganglion cells, which are the output neurons of the retina, and this particular type is sensitive to directional motion stimuli. So these ganglion cells only react if a certain direction is, or the bar that stimulates it has a certain direction. And they were identified first by light microscopy. This was calcium-based imaging. And then we used the exact same tissue block and put it into the electron microscope and imaged it with a method that I showed you. These were, by the way, 13,000 sections taken automatically over a time of 6 weeks. And then by correlating the light microscopic picture with the EM picture, we found the very cells that we had functionally identified and could then, using our reconstruction tools, reconstruct those neurons, particularly the ones that we knew functional properties of. So we didn't have to do the dense reconstruction, we just chose those that we knew something about. And here again, I think you can nicely see the complexity of what we're talking about here if you want to reconstruct neural pulse. So these are the few neurites that belong to that very cell. All the rest are all the other hundreds of neurons in the same data set. And if we fly a little bit further, already now you have no way of seeing single processes anymore because the scales are just so enormous. And now comes out basically the reconstruction of this particular ganglion cell, again, for which we knew the functional properties. And if you turn it around, you see that it's a nicely actually bistratified cell that is known to exist in the retina. So this is now the light axis here along the y-axis, and this is the inner plexiform layer in the retina where these cells are known to stratify. The question we then had was whether we could use this method now to identify specific presynaptic inputs to these cells that would explain their directional selectivity. And the hypothesis had been that a certain type of amacrine cells, one of them is shown here in blue, would make specific directionally oriented connections to these ganglion cells, and because of that directionality of the inhibitory input, you would create the postsynaptic cell's functional property. To test that, we looked at the output synapses of those starburst amacrine cells that we reconstructed in the volume also, and labeled the synapses, those synapses that were made with one of the ganglion cells we had recorded from. And the color of the labeling here corresponds to the functional property of the postsynaptic cell. So again, we have a presynaptic neuron, we look at the synapses of that presynaptic neuron, and label the synapses by the functional property of the postsynaptic cell. Question being, is the wiring of that presynaptic cell specific for what the postsynaptic cell does? And the simple color code here already shows you that the answer turned out to be yes. So for single cells, but also in the average here for the entire population, you can see that there's a nice, if you want, color clustering here, which means that the dendrites of single starburst amacrine cells decide by the functional property of the postsynaptic cell whether they make a synapse or not. This is seen more explicitly in the view from the postsynaptic cell. So if I can focus your attention on these polar diagrams here, you typically represent a functional preference in such polar diagrams where in color here, these are the functional preferences of the ganglion cells we measured. However, now with our EM method, we could add the structural tuning curve, if you want, which is the directionality of the input dendrites coming from the starburst amacrine cells. And as you can nicely see, most of these histograms nicely align basically in opposite directions, 180 degrees between each other. So the functional preference goes in one direction, the structural directionality is exactly opposite. And this makes sense because the synapses are inhibitory. And now if you look at these combinations of structural and functional tuning curves, we think in this particular case we could invert the argument and say that actually this is likely to be the cause of the directionality of the postsynaptic cell, that actually the inhibitory wiring is so specific that it allows to create the functional preference of the postsynaptic cell. So this was one example where we took a reasonably large volume, reconstructed a specific subset of cells, only 30 in this case, to answer a specific question, which was possible because we knew something about some of the functional properties of these cells. The second example now finally gets at dense reconstruction, the actual, if you want, claim of connectomics, which is get at matrices of connectivity. And for that, we used a different tissue block, also from the retina of the mouse, sized a little bit differently. It's 120 microns, roughly on the side, times 80 in this plane. But it spans the entire retina all the way to the photoreceptors here. So these are ganglion cells and here photoreceptors. So what did we do with this tissue block? First, we wanted to just get a soma map, a map of the cell bodies in that volume, which you can see in a second. So this is just basically a first order map where we know where the cell bodies are. And this is ganglion cells on the right again, and then here in a nuclear layer, it's blue is amacrine cells, red is bipolar cells. Then out here you have the photoreceptors. Well, that's just cell bodies. That's not so super interesting. We wanted to get at the neurons reconstructions. For that, we used a large tracing team, which at the time was 50 people. Now it's 200. It's undergrads in Heidelberg and Munich, and they actually now did the job of converting the soma map into real reconstruction. And these are now all the bipolar cells in that particular volume, 460 of them, which already was kind of nice to get. So the color code here now is by the stratification of those bipolar cells. This is just an example of a morphological identification of certain cell types, and we can walk through them. They're nicely laminated as expected, and we find the bipolar cell types that were expected to exist. And this is just a movie flying into this, so from the distance it seems pretty dense, but if you fly closer, then you will notice that first of all, we generated these skeleton reconstructions. As you can now see that each of the neurons is only kind of a thin line in the reconstruction. We'll get to that later. And also you can see there's still quite some empty space, so this is only a subset of the neurons, although it's so many, and although it looks so dense, again an example of the density of this kind of problem. Okay, we also reconstructed the ganglion cells, all of them. There are different types, some of them known, some of them less known, and went also for the endocrine cells. So this totals to close to 900 neurons that we reconstructed in this particular one tissue block. Just to relate it a little bit to the literature, you can then look for types that have been described, rod bipolar cells, for instance, other kinds of color-sensitive bipolar cells, or off bipolar cells. You can ask whether the cells you have form a mosaic because they represent the visual world in a mutually exclusive manner, most of them at least, and you can get these nice dendritic and axonal mosaics. And that's the kind of reconstruction we get. We have ganglion cells, as I said, some of them described before, by genetic markers, and so we know that basically the cells we have are reasonable for retina specialists. We also found a cell type that was not existing or was not described in the canonical form before, but that's maybe more interesting for the retina specialists. We also went and reconstructed all the endocrine cells just to show you the variety and the variability of neuronal form and the density again of that kind of reconstruction challenge. Beautiful, asymmetric endocrine cells, bistratified endocrine cells. Okay, that's just cells, and cell types is interesting, as Jeff Lichtman pointed out, but it's not the end of the story. Of course, we want to get at connectivity. So how can we now finally get a matrix out of this? This is the last thing I want to show. We combine skeletons and computer output. So skeletons do not allow us to talk about proximity of neurons, and especially not allow us to detect touches between neurons and analyze the geometrics of touch. This is possible if we grow it out into volume reconstructions, and this we do by using the skeletons as seeds for watershed and more complicated transforms to get to the volume reconstruction. This is shown here. This is a skeleton. This is what happens if you combine all the objects that the segmentation generated that overlap with that skeleton. I should say that these segmentations were generated based on classifications that were trained by Sebastian Sung's group at MIT. And so this is what happened if you only had the automated output, but then because we have the skeleton, we know that these belong together, and then we can take a pair of cells and ask whether they make contacts, as shown here. So there's a nice bipolar cell contact to a ganglion cell, and then there's another one there, and can analyze that for each of the cell pairs. Substantial detail. This is another example of a bipolar cell touching a ganglion cell, where then we can measure the surface area properties or the contact area properties and use that to detect what is likely to be a synaptic contact. And then, as a result of this, we have generated this attempt at a connectivity matrix of a piece of retina. It has the size roughly 900 times 900 because that's the number of neurons we had in that tissue block. And the color code here that is hardly visible, but maybe a little bit, is now the likelihood of synaptic connection, and is an example of where we actually wanted to get, which is really take a block of tissue and map these connectivity matrices. And just for comparison, the C. elegans connectome that had been worked on in the end of the last century contained 300 cells. So this is now, it took 15 years, and this is now an example of a circuit or a matrix roughly 10 times the size, and it took us 2 years. So if you add methods development, more like 4 years, but it would take you today maybe less than a year to redo it. So this is really now the improvement that has come out of this. Just to get used a little bit to how to look at this matrix. So if you take a piece of it, then each of the entries is a cell pair, and each of the cell pairs has potentially multiple contacts. So there's a lot of information in each of these entries about typically tens of contacts between a cell and another cell, and others don't contact, like the white and the red one here. So this is the kind of information that we have in there. We can then use that to, for instance, classify types of cells based on their connectivity, which we did for a certain bipolar cell type. Just want to show you some of the pictures here. So we take a set of cells that has not been able to distinguish, but connectivity actually pulls apart these kinds of cells. And that's just one example of how to use this. Another one is to use a cell type that we had not seen before, that giant bipolar cell, and ask what the particular circuits of that cell are. Then you can walk through and basically pull out the cells involved in that circuit, and then really walk through your matrix and make your kind of connectivity analysis, ask what ganglion cells are involved in that circuit, and play that game over and over, which is really a nice tool now, because instead of doing each of these experiments in a new project, we can basically take the matrix once we have it and now ask all of these circuit questions one after the other, having the matrix at hand. And yeah, that's just an example of how we've pulled out circuits of this very sparse giant bipolar cell, a very rare cell, but we can now basically draw these circuit diagrams based on this data, and this is just one of the many circuits in that tissue. Okay, so to summarize, I've introduced the techniques we are using for what is nowadays called connectomics, or we prefer the term structural neurobiology because it nicely makes the analogy to structural biology and the challenges there. We're using serial block phase EM, as I showed, and I try to make the point that reconstruction is the bottleneck and indicate how we're using crowdsourcing and machine learning to solve this. And then I showed the first kind of dense retina reconstruction that we obtained. I want to thank the people involved. This work was done with Winfried Denk and Kevin Brickman at the MPI in Heidelberg. And then, of course, I want to thank the hardworking tracers who are reconstructing as we speak new stuff. I just want to give an outlook, because this is next generation conference here. So what we're doing next is not retina anymore, but cortex, because that's, I think, where really the true miracles of the brain lie. This is what we are doing in my group in Munich now. We pick a tissue of cortex, 4,000 neurons, and reconstruct it, asking what the structure of the matrices are in primary sensory cortices, trying to then infer how actually pattern recognition, which computers are not yet good at, can be done in a cortex. And just because I want to give you some novel thing here. So we're working on computer games to help us reconstruct, and this is just a prototype that we recently built. So these are browser-based games with the idea that we can scale up reconstruction even more by asking people to help us and not just pay them for it, just as a brief outlook where the challenges actually lie. So thank you very much for your attention.
Time for a few questions.
Can you distinguish between just a happenstance proximity and what actually a synaptic connection? Are there fixed areas or something that you actually see? Distribution of areas?
Yeah. So the answer is, if you had only one touch, it's reasonably difficult, but we have two solutions to that. One is some synaptic contacts are very conspicuous, and in the case of the directional selectivity circuit that I showed, the contacts are really like wrappings of the postsynaptic dendrite, and this is so conspicuous that we could use just this geometrical feature to detect synapses. More generally speaking, we need training data to answer that question, and therefore we've also taken data where we have a labeling of all the vesicles and everything you need to actually detect synapses, like shown here on the right-hand side, and we're using these to calibrate geometrical features of how a synapse looks like. That is the case for the retina. In the cortex now we're just taking large amounts of this kind of data where we have all the vesicles and postsynaptic density stained, and then the problem is solved by that.
Can you give a sense of like the amount of data in terabytes that the retina, the full retina connectivity matrix, not just the matrix but the structural data itself, like how large is all of that?
So the data set we obtained for the directional selectivity question was 1/3 of a terabyte. The other data set I showed is roughly 1/2 of a terabyte. So that was still just the retina. For the cortex, what we're doing now, we're talking about, or the data set we want to acquire is 30 terabytes, so 30. So that's the scale, and I think we and others have worked on improving imaging speed, and that will allow us to typically get dozens of terabyte-sized data sets. So that's the scale, but again, I mean, the longer the field runs, the quicker it will be, and Jeff Lichtman introduced this multi-beam system, which we're also working on, and this will be, yeah, as you said, I mean, gigabytes per millisecond, basically.
Moritz, having a
complete wiring diagram would be great. I'm wondering about the steps, sort of results that you can get before you get there, before you get to the complete stage. And in particular, I mean, these connection matrices, these classes of connection matrices, the synchrotron chain, the law, etc., will not require anything like a complete wiring diagram in order to evaluate which of this is true. So I'm wondering, to what extent are you targeting these sort of intermediate results?
We absolutely are. So there are two ways of kind of subsampling that question. One is, do you actually want to start with smaller volumes? And that is a little bit challenging because, as I showed, many cells are very large, so that is more difficult. If you go too small, then you just never have a complete cell at all, and that gets difficult. However, let's say the imaging works anyway, because the reconstruction is the bottleneck, as I said. Let's say you have the volume. Then, of course, targeting your reconstruction smartly is important. So, for instance, if you start with inhibitory neurons, which are also very interesting, but only make up 10% of the cells, already a big step. You can find out about all of their wiring. You can start with dendrites, which are, again, only 10% of the wires. Again, you have already a complete set of those and can look at it more specifically with respect to this connectivity matrix. It depends on the hypothesis about the structure, of course, what you have to sample, and that is something that we and others are working on to basically write down, assume you want to find, or your hypothesis is this kind of circuit, what is the density of sampling you need to distinguish that from another circuit model. And of course that is not necessarily complete unless structures are maybe different from what we expect, and none of those hypotheses may be true, and then of course the more we have, the more we know.
Okay, thank you.